identification and the generally accepted rules of taxonomy if you're interested in doing some field work. You'll get an idea of how to ID lichens both in the field and in the lab, and I'll share some of my favorite resources around that. And then by focusing in on the words and names that we give these organisms, we'll unpack their interpretive capacity for naturalists. And we're also going to attempt to settle some of the complexities in identification that often shut out laymen from studying them. So with the technical stuff out of the way, then I'll kind of give an overview of human relationships and our ethnobotanical uses for lichens, their ecological niche, and I'll start to kind of open the space for their interpretive potential. So the goal of this presentation is to show a beautiful array of entry points into how to study them and maybe seed a little bit of curiosity in you so you can find what about them is the most interesting. And hopefully, maybe it'll blow your mind a little bit. If anyone at any point in time has a question, feel free to drop it in the chat. I can try to address it as I go, or I might circle back to them at the end. All right, so in order to teach you about lichens, I'm going to have to teach you evolution again. So in the very beginnings of the study of the origin of life, lichens were in the room every single time. And this was the most surprising thing I learned when studying lichens, was that they were at the crux of all of these major issues in our very conceptions of what life means or how life came to be, and yet they weren't considered particularly important to science. So to begin, I have to begin all the way back with Charles Darwin's grandfather, Erasmus. The study of lichen has taken some relational turns over the years. So I'm going to try to kind of track it for you and show you the ways that it's sort of gone in circles. So Charles Darwin's grandfather, Erasmus, shared a hypothesis of something called transmutation of species in the 1790s, in which he believed that organisms formed spontaneously and then used and disused traits. And that's essentially how they dictated their morphology. Meanwhile, in the 1790s, a student of Linnaeus named Eric Acarius continued in Linnaeus's legacy of being the first taxonomist, and began classifying an essentially undescribed category of lichens, trying to classify them for the very first time into subdivided genera. He made incredible drawings of lichens, and these are the first written descriptions that we have of lichens in a scientific way. He collects over 5,000 specimens in his lifetime and becomes world-renowned. And in 1819 in Spain, he died while looking at some lichens on a tree. Then it takes about 40 years for anyone to circle back to anything that really relates to lichens. You'll find that the study of lichens is essentially maintained by one scientist at a time. We're hoping that that's going to change in the near future. But for now, it's how it is. So 40 years later, Darwin publishes The Origin of Species, in which, as most of us know, he essentially lays the groundwork for evolutionary theory. While at the time he received significant pushback from the scientific community, and in particular the Church of England, because this idea of his grandfather's of spontaneous formation and his idea of one common ancestor are fundamentally different in the story of life, very different origin stories. His visualization of the branching trees of evolution became core to our understanding for the last 150 years. Then, 7 years after The Origin of Species essentially rocks the world, Ernst Haeckel has been making some wild and very poetic observations about the relationality of life, and he coined the term ecology at that time. He believed that organisms were relational at their core, and he could not be understood in isolation from their environments or the company that they keep, or in his understanding, organisms were what they were surrounded by. At this time, lichens were understood as discrete singular species. So any interested party would have looked at it and identified it as an individual, the way that we might with a squirrel or a tree. But Ernst Haeckel begins to lay the groundwork for more radical conceptions of what we believe is an organism proper. He produces incredible drawings, still are some of the best drawings of lichens that we have. He believed they were comprised of two vastly different organisms. This is the first person to observe this in the lab. A fungus that acted as a parasite, he believed, and yet also a tyrant in control, and algae cells that were intertwined, that were essentially slaves to the desire of the fungus. This was widely opposed by naturalists at the time, primarily because there was no real precedent for an interrelated body of organisms being called one thing, especially a pairing that forms a creature that's completely unrecognizable from its parts. Taxonomists were rightly concerned and at times infuriated by this concept. It made it very difficult to categorize based on how science was shaping up to be. Plants and fungi were also in completely different taxa of life. Rather than making order out of life with clear lines of descent flowing down from one root, the way that Darwin's concept dictates, this hypothesis insinuated that species could be emergent rather than divergent. And it kind of feels like Charles Darwin's grandfather Erasmus would have actually preferred this concept. Even those scientists that eventually came around to the dual hypothesis began to disagree with its phrasing and the relationship that he described, the kind of master-slave dynamic. That narrative would be eventually tabled for a now beloved term, symbiosis. In 1877, Simon Schindiner puts forth his dual hypothesis. So taxonomists are angry. This is a subset of science that is essentially being widely silenced because it doesn't really go along with what they're willing to absorb. So then we have to jump forward to 1877. The word symbiosis is coined for the first time to describe a mutualistic relationship between lichens. So lichens are in the room for essentially every major scientific discovery of the last 150 years. In 1878, 1 year later, a German mycologist defined it as the living together of unlike organisms. This definition has been hotly debated, with some scientists advocating that it should only refer to persistent mutualisms, while others think it should apply to all biological interactions, including things like parasitism or commensalism, or even brief interactions like predator-prey. Regardless, we owe a very important biological concept to the study of lichens. Symbiosis is used extremely widely, and it was observed first in the lab as a lichen. The focus on this verbiage is starting to shift from something highly competitive to something that's naturally collaborative. Rather than master-slave, we have symbiosis. It took about 60 years. In 1909, a Russian lichenologist presented a research paper, The Theory of Two Plasms as a Basis of Symbiogenesis, a new study on the origin of organisms. Kind of a mouthful, also kind of a crazy person. He aimed to explain a new theory of evolutionary merging at the core. So these new ideas can be studied today under the title of endosymbiosis. An endosymbiont or endobiont is an organism that lives within the body or cells of another organism. So this is now considered extraordinarily common. At the time, this was seen as insane. Typically, the two organisms are in a mutualistic relationship. Examples are nitrogen-fixing bacteria, which live on root nodules of legume plants, single-cell algae inside of reef-building corals, and bacterial endosymbionts that provide essential nutrients to insects inside of insects. So we're seeing the dual hypothesis theory propel thinkers into this kind of uncharted territory around the relationality of organisms. However, the dual nature of lichens was no more than a theory until 1939, so almost 100 years later, when it was able to be proven by a Swiss researcher. And he was able to reproduce in the laboratory the phenotype of the lichen Cladonia by combining two of its identified components, a very beautiful cup lichen. It looks very different from its parts. The first successful lab synthesis of lichens, pretty impressive. Even today, that is a very difficult task. Lichens tend to be a little bit moody. So even if you put them in the perfect circumstances with the perfect substrate and the perfect ratio, we still don't know if we can predict whether or not they will lichenize properly. You can see here a specimen that he created. It's extremely beautiful and somewhat common in North America. So now we are jumping ahead. about 30 years to the 60s, a somewhat radical time, if any of you remember, in which these theories might be a little bit more easily absorbed. Lynn Margulies proposed that some of the most important moments in evolution were a coming together and a staying together of different organisms, that the mitochondria in our cells and the chloroplasts in plant cells were once single cells or bacteria that were swallowed by larger cells. She calls this the intimacy of strangers. People said that Lynn Margulies was a crazy dreamer. However, in 1978, she was actually proven correct. Weathering consistent criticism of her ideas for decades, Margulies was famous for her tenacity in pushing her theory forward despite the opposition she faced. The descent of mitochondria from bacteria and of chloroplasts from cyanobacteria was experimentally demonstrated in 1978. So this formed the first experimental evidence for the symbiogenesis theory, which had been run for easily 100 years by then. The endosymbiosis theory became widely accepted in the 1980s after the genetic material of mitochondria and chloroplasts had been found to be significantly different than that of the symbiote's nuclear DNA. So there were cells within cells that were genetically completely and fundamentally different. The endosymbiotic theory literally rewrote the history of life. Lynn Margulies was an absolute hero in her time, and she is biology's unruly earth mother. Lynn said of lichens, they detail the complexities of individuality, which I find extremely beautiful. Meanwhile, Joshua Lederberg is born. He's extremely young, but he's considered a prodigy. And he won the Nobel Prize at 33 years old for his articulation of something called horizontal gene transfer. This is a concept that greatly disrupts evolutionary theory, similar to how the dual hypothesis had. He found that bacterium could trade genes with one another, picking up traits sideways as they went through the world that would alter their genomic structure. This would be the equivalent of me putting my arm on a stranger next to me on the bus and absorbing her hair color or the texture of her skin. Nowadays, this is completely normal in science, but at the time it sounded unbelievable. Prior to this discovery, bacteria and all life forms were essentially considered biological islands, completely separate from one another. Nowadays, horizontal gene transfer is mapped across time, and we no longer consider organisms to be fully discrete, closed systems like we once could. This is also due to the rise of microscopic visualization technology, the microscope, but also a camera that can see on a cellular level. Now that we have these, as well as the profusion of genetic testing, we're able to disprove certain theories of science which were set in stone. So now that we know that nothing is a fully discrete closed system, the tree of life is becoming an immense web in which traits are passed across all domains of life, and all domains of life cohabitate within singular bodies. Lederberg illustrates convergence as the absolute norm, and in 2001, he coined the popularized term microbiome. So the idea that all of the bacteria in your gut are completely different genetically from you. However, you live in total symbiosis with them and you're actually dependent upon them. And we know that the microbiome is passed from mother to child during pregnancy. So these are some really foundational changes in the study of life that haven't quite made it to the American textbook yet. In 2016, thankfully, we have proof of the multiplicity of lichens. So rather than just being a algae and a fungus or a cyanobacteria and a fungus, in recent history, we have discovered that lichens are nothing like that at all. A group of researchers in Montana were challenged by a friend to grind up lichens and sequence their DNA. Expecting two dominant fungal and algal partners, they were shocked to find a wide array of organisms, wider than they ever could have imagined. He tested over and over again, attempting to control for contaminants that he was receiving in the tests, eventually realizing that every single lichen was not only host to two bionts, but to a wide variety of interested microbes that were actually stable partners in the symbiosis. The lichens almost conceptually resembled an ecosystem. As it turns out, he and his team have yet to find a lichen that fits the definition of one fungus and one alga partnership. So in this image that you see on the slide, those two lichens are morphologically extremely similar, the only difference really being color. But the difference genetically is actually a vagrant yeast. Vagrant lichens are microscopic. They are so lightweight that they can float through the atmosphere, and they usually live pretty high up in the atmosphere as well. So they tend to travel down in rain droplets or with storm systems. And those vagrant yeasts will partner with fungus and algae and fundamentally change the morphology of a lichen. It seems like a really crazy process, and it's not really something that we've ever witnessed, but it can speak to some of the problems that we've had with creating lichens in the lab. Perhaps there were partners that we just had never even seen before, had never been able to trace. So there are still significant mysteries with lichens, and this is an understudied area. Okay, so now that we've established that everything that you were taught is different now, and everything is made of everything else, and there are no boundaries to anything, we can move on. How do we deduce meaning from something that's so complicated scientifically? Um, there are really awesome resources on lichen philosophies. There's a couple of incredible nerds in the Pacific Northwest that have done amazing philosophical work around this, but have also done a good job of mapping all of the different scientific studies and the kind of perspectives that they come from. So in this portion of the talk, I'm going to talk about the kind of dynamic philosophies. There are many different ways, 8 primary ways to define lichens, but how you think of them is essentially what they are, as science has shown over the last couple of hundred years. So what are they really? Um, at this time, I think the fun game is to pick your favorite interpretive understanding and to recognize that there are times and places for every interpretation of lichens. I think that a good ecologist knows how to use their senses in a given environment. So it's sort of fun to use this as a thought experiment and to approach the same lichen, but from different perspectives to try to see how it might have formed. So lichens are kind of mirrors for our own conceptual bounds. They're sort of like portals, if that makes sense. So we'll start with what lichenologists are primarily engaged with today, which is making sense of the different constitutive partners of lichens. What are they made of? And trying to deduce that constitutionality. And these perspectives are pretty set in stone. The first few are constitutive. So lichens are either fungus-induced constructs or they're algae-induced constructs. This is the perspective that is the basis of most studies, I would say. It's focused on running of genetic tests, gathering of genetic materials, sorting out all the mess, and then making sense of who's in charge, essentially. And so the definition of the lichen is the most prominent partner in that lichen, usually the fungus. So in this concept, lichens are kind of about the fungal partner. And then the algocentric view is a little bit more subversive. The only people who are really into that are ocean studies people, people who like cyanobacteria. And it's sort of about how lichens are the algae. So this is kind of an interesting dynamic. Is the algae being farmed or is the algae the farmer? And then there are relational perspectives, not that the lichens are what they're made of, but that the lichens are their relationships. So the symbiotic perspective is pretty common, I would say. It relates as a fungal algal construct, essentially. It's sort of a process-oriented view. Ecologists tend to really like this one because symbiosis is a pretty clean concept. And it sort of treats lichens as a consortium of differently named organisms that are in cost-benefit relationships with one another that we can basically map the way we would map an ecology system. And then there's an ecosystemic perspective, which is also highly relational, but is a little bit more overt in terms of it being ecological in nature. So lichens are mini ecosystems in this idea. They interact as complex systems and they give rise to nonlinear relationships and emergent properties. There is very little research on this conceptually, even though to me it seems pretty obvious. Now that we know that lichens are made of many, many things, why wouldn't we be able to see them the way that we see a woodland forest, for example? So patterns of carbon cycling in macrolichen thalli are fractal in this concept, and they can be interpreted with ecology principles. I think this is an area of study that's pretty thrilling and a little bit more intuitive than some of the harder scientific forms of study of lichen that are popularized today. And then we have something a little bit more complex, like a phylogenetic perspective, an evolutionary perspective. So they're essentially considered evolutionary termini with their own respective lineages that have merged for situational survival. So the microscopic kind of molecular scale is interesting, and the time scale is pretty ancient on this too. So it's not like they emerged because there was a given circumstances, a given weather event, a certain kind of environment they were in. This is a process of emergence that's tracked as pretty ancient. Um, so it's still pretty traditional in the sense that it's about what they're made of. It's piecing them into different pieces rather than seeing it as a constitutive whole. However, that's because it's showing independent lineages that form different sets of relationships. So you're probably thinking this is getting a little convoluted, and it's true. It really is. These sort of micro analysis moments really show you how your perception of how to study something can actually dictate the knowledge that you find in that thing. So this philosophical work can go a long way, especially for lichenologists and people that study fungi, because it might give them an opportunity to see a different window into the data than they would have seen prior. Then there's a sort of organismic perspective, which is sort of fun. It's a more emergent kind of concept. It's also rooted in symbiosis, but it describes the lichen thallus as an emergent entity. So it's helpfully understood in terms of their interactions than of independent evolutions. This is kind of fun because it's sort of like you can tell that scientists are kind of arguing about what the most important thing to look at is, when the truth is, it's all important. And then we have organismic perspectives. So this is sort of intuitive for naturalists, I find. They kind of like this one. The lichen is a unit of internal physiologic response to its environment. This is a way of thinking that ecologists quite like. So lots of research relates to this concept. They operate within a single unified and unifying physiology. Each little part of the lichen has a different role. And the relation between the form, like the way that it looks, the morphology, can be empirically elucidated from the inside out. The morphologic perspective is the opposite. The relation between lichen forms, functions, distributional ecologies, and even constitutive partners can be inferred from the outside in. This is a fun game. There's a lot of science on it. However, I would say that it's a stretch. It's a stretch. It's highly interpretive in nature, and unless you're studying them on the most microscopic levels over many, many years, it can be hard to make these kind of generalizations. So now that we've gotten the chunky stuff out of the way, it's clear that there are many different ways to understand what a lichen actually is. But I think I'd like to zoom out and kind of speak on what they do in their ecosystems. Lichens are essentially the first sign of life when nothing else can survive. They're a pioneer species. So after incisive events such as floods or especially fires, they're the very first thing to grow. This is likely because their constitutive elements exist in the ecosystem essentially ubiquitously. They're everywhere, as well as the concept of vagrant yeast I was speaking to earlier. Lichens can quite literally float in from outer space. They are also a sort of bridge between the living and non-living world. They do a lot of work on the edge of life in order to support the rest of the ecosystem. So their fungal hyphae, the fungal element, can grow several millimeters down into rock. It's extraordinarily strong. It can push along cleavage lines and carve through rock. They produce substances called depsides that chemically combine with minerals that they catch from the air to create metal complexes, which actually make rock more soluble. And they're able to do that in a way that's fundamentally different from plants because they have this multiplicitous capacity. They're doing the work of a fungus, but they're also photosynthesizing simultaneously, and they can respirate minerals from the air. So the weathered rock that they're creating is the beginning of soil. We wouldn't have bioavailable soil for plant life if it weren't for lichens. Some acids that they produce are extremely weak in water, so the formation of soil might take centuries. But some, such as carbonic acid, which is commonly produced by lichens in the hill country, can eat away at rocks that are rich in calcium carbonate, like limestone, relatively swiftly. Um, when lichens die in place, they are the first layer of soil. They trap dust and silt. They accumulate all kinds of microbial life, and they wait for a humble moss spore or for a seedling. So if you've ever seen a tree absolutely covered in moss, you can almost guarantee that there was a lichen before it. They are pioneers because they can withstand quite a bit. They can withstand drought for over a year. They can completely, essentially, they can eject all of the water out of their system. So they don't experience cellular death the way that plants might if they were being roasted in the sun over time. They also have built-in UV and radiation protections that are quite strong. Certain lichens have melanized to the point of being able to survive extraordinary high levels of radiation. And that's still an understudied area, but in the lab has been pretty remarkable, the lengths that they can go in terms of protecting themselves from UV. Um, they are in a strange way also self-sufficient, which is what makes them a pioneer species. They produce all of their own carbohydrates and sugars. They gather traces from atmospheric mists that they receive of minerals. They also mediate the deficits in a given environment. So for example, some lichens have a partner that is cephalodia, which is a kind of cyanobacteria, like a blue-green algae. They tend to colonize substrates that are nitrogen poor, and they can become nitrogen fixers essentially overnight, creating their own from literal thin air. They cannot compete with things that grow quickly, but they can compete with the rawest elements on Earth. They consolidate and stabilize soils, they become organic matter, and even their color changes and reflectivity of light can change water retention in the soil. So this is widely acknowledged by foresters in Quebec in the healing of the boreal forest after fires. Lichens actually change the color of the soil from a dark, dark black to a beautiful light blue, and that reflection does a lot in the ecosystem. So even down to their color, they're actually serving the whole of life at the core. Right. The colonization of trees is its own kind of special section. So where the succession of lichens on rocks is pretty obvious, they start kind of small and crusty and become increasingly more like foliage and eventually give way to plants. The succession of lichens on trees is very complex. It's affected by the age of the tree, the chemistry on the bark, the moisture holding properties of that bark, and the weather. So lichens are of huge importance in forest ecosystems. They harvest ambient minerals from fog, like I've said. They absorb nutrients and then release them down in concentrations below. So they're always sort of flaking off and regenerating the soil. But they're also able to do things for the bark of the tree. This is an understudied area that's quite interesting. Around here, we have a lot of cedar trees, for example. There are certain species of tiny, tiny, tiny white lichens that will only grow on very young cedar trees, a cedar tree that's under 5 years old or so. And then as cedars age, they go through probably a 15, 20-year period where you're not going to see any lichens on that tree. It's growing too quickly. The acidity of the bark is too high. However, if a piece of that tree were to die, it would immediately be colonized by completely different lichens than had existed on it in its prior form in the life cycle. So there is a lot of mystery related to this, and it's a very understudied area, but it seems like trees go through fluctuations or changes over their lifetimes that can be aided by lichens. And additionally, we know in the hill country that lichens do an extraordinary job of cooling the core temperature of a tree. So they aren't parasitic at all. They actually increase surface area, hold water on the outside of the bark, and prevent the tree from heating to the core. Um, so most important in the forest ecosystem is the contribution of fixed nitrogen. Nitrogen in the air can only be integrated into living systems by converting it into nitrates or ammonium compounds, and lichens do this extraordinarily well. Only a select few organisms can do this, and it's primarily cyanobacteria. So their pattern of hydrating themselves fully and then drying out and going dormant has been shown to impact the level of humidity in the ecosystem. This is an area that has been studied quite a bit up in the Pacific Northwest, where rainforests are heavily studied, as well as the sort of talk around atmospheric rivers or the ability for trees to breathe and respirate air at their choosing. Lichens are sort of in that conversation. However, I've yet to really meet anybody in Texas who's integrating those concepts into our forest ecosystems here. So I think that there is a lot of room to be looking into what lichens are doing in terms of humidity. And in the hill country in particular, we have a kind of foggy, kind of misty season through the winter that is absolutely defines the region. And this is prime lichenizing times. And it feels like in the early morning, they're breathing on you or something. It's a very tangible feeling when they've opened up and they're actually doing the biological processes that they've been waiting for all summer. I see in the chat, is the lichen actually colonizing the tree then, or is the tree one of the partners in the lichen? That's a wonderful question. I think in certain understandings, the tree is in a way a partner. The tree is like a habitat for a habitat for habitat. However, I would say that because the tree and the lichen are not we have yet to prove that they're fully exchanging. So we're proving that they're bound to each other. We're proving there's a little bit of mineral tapping coming from the lichen, which is why the word colonization is commonly used. But we have yet to show whether or not the lichens are giving anything back to the tree besides a kind of like they're not giving back anything chemical, they're giving back material and they're giving back shade basically to the tree's bark. Um, but I love your thinking in that we can really crack open the edge of the organism. And once you do, it's really hard to stop doing. So it's exciting to imagine that maybe trees are partners to lichen, or maybe everything growing next to one another are partners at the core. The one lichen tree relationship that I have seen is the lichens grown on beech trees. Absolutely. That is definitional to a beech tree. When you imagine a beech, you're actually imagining the lichens on them. So pretty wonderful example. Thank you for that. So a lot of people look at lichens and guess that they are parasitic. Really common mistake made around here. Or if not actively, they are at least passively harming the tree. There is no strong evidence of this whatsoever, and they essentially don't grow deep enough into the vascular system of the tree to ever interrupt its functionality. So they are, however, impacted by loss of leaves. They tend to thrive when there are openings in the canopy. So any exposure of the bark to more light is going to stimulate the growth of lichens. So people often make the mistake where they see an opening in the canopy, they see a bare branch with no leaves on it, and they see a lot of lichens on that branch. That is usually the first branch to be chopped and mulched and sent away. However, that is your tree responding to its environment, likely losing leaves for some kind of good reason, and biodiversity steps in to make use of it. So if you have trees with dead looking limbs that are covered in lichens, I would recommend keeping them as long as possible and then letting them fall where they lie is probably a great idea. It's a good way to nourish the tree and to nourish the ecosystem around you, and they will regenerate and form lichens in other areas, which is a net positive for the environment. Um, so another sort of causation correlation fallacy is that dead trees do not grow and lichens prefer very stable environments. So you'll see lichens on dead trees primarily because it's static, not because they have killed the tree. That's also why we don't have an abundance of lichens on cedar. They're extraordinarily fast growing when they're in their teen years. So lichens tend to stay away. Um, the growth of foliose lichens, the big fluffies, is technically a home for insects. So there's certain examples where somewhat harmful moths that defoliate trees like to live inside of lichens. It's called the lichen moth, but that's in Canada and doesn't really apply here. But they also tend to be habitat and food for many creatures, big and small. If you spend more than 15 minutes looking at a singular lichen colony, you will notice it is absolutely teeming with life, much of which you can't really see unless you sit and stay for a while. Um, but my favorite part of their role in the ecosystem is that they're beautiful nest material for hummingbirds, and many different creatures use them in different ways. One of the famous ones is reindeer eat lichen. It's sort of their favorite delicacy. When the permafrost is really dense, they can dig up lichens underneath that are surviving, and it's a great way to get nutrients in the wintertime. So now I'm going to kind of talk about the human uses. This is sort of a fun one because they're a little bit wackier than people realize. So there's a practice, kind of old, but it still applies. It's called lichenometry. So you can date rock surfaces, you can date gravestones based on the change in lichens. So what you need is an old photo of a gravestone from 100 years ago. And then you can track that change, and based on pretty simple now AI integrated technology, you can basically guess the age of that rock that it's sitting on and how old the lichen is. Um, there's also a fundamental need for the study of lichens as they relate to air quality. Lichens are absolute bioindicators of the health of an environment. So if you're in an area with very low air quality, the likelihood is you're going to see some small crusted lichens on concrete, but you're not going to see much else on trees. This is because they are very sensitive to air. They're constantly mineralizing and respirating things from the air. Lichens have been shown to absorb pretty horrible chemicals from the air. So they're doing great work, but they can't sustain it for particularly long. There are some long-running air quality surveys in the US, and in fact, the longest running one is a lichen survey. So it's pretty exciting. Um, We've known that they're sensitive to air pollution for about 150 years because in initial tests, we were able to find a lot of air quality toxins and pollutants inside of them. So they can absorb components of acid rain, fluorides, they absorb ozone, hydrocarbons, copper, lead, and zinc. And they can even absorb sulfur dioxide that comes out of cars and vehicles. And each of these impacts the growth of lichens in different ways. So the really good lichenologists can tell the difference. from a lichen that has absorbed sulfur dioxide in high amounts or a lichen that's absorbed zinc in high amounts, for example. Some are a lot more sensitive than others, you'll find, as evidenced by the proliferation of urban crusty lichens and the complete lack of foliose lichens in a city. So by measuring distributions in somewhat wilder places and comparing this to city data, a picture can begin to form of the flows of certain pollutants. You can actually map pollutants in the air based on lichen surveys. And in England, they have created a 10-point scale system that can be directly related to average atmospheric sulfur dioxide, and it measures differential sensitivity in lichens. And the system is so simple that it can be taught to school children in England, Ireland, and Wales. So this is a pretty common thing that school children there will experience in public schools. And we can actually create citizen scientific brigades in about a 2-hour session. Um, so when surveys are repeated over and over against time, you can map pollution directly. The best example of this is an inventory of lichens at the Indiana Dunes State Park in Lake Michigan that's near Chicago. Pretty close to Gary, Indiana. It's a highly industrial zone. So we have good lichen studies from about 100 years ago in that region. So the study was recreated, and he compared it and found that the disappearance of lichen flora was at 80% within 95 years. So that's a direct correlation with the industrial build out of Lake Michigan. So it stands that lichens in places with new restrictions on pollution tend to recolonize. So if an area becomes suddenly a lot less polluted than it was before, you're likely to see a recolonization event happen. They're considered essentially the canaries in the coal mine in spaces of ecosystem protection. And the Forest Service and National Park Service now have baseline lichen studies when they acquire new land. So as bioindicators, this is probably the most useful they are to human beings. And now I'm going to talk a little bit about ethnobotany. There aren't a ton of cultural linkages to lichen. However, certain cultures, they were way more charismatic. And especially in northern and Arctic cultures, this tracks to me because when the trees lose their leaves is when lichens look the best. And they can survive pretty well in very cold environments. Usually they're not considered a primary foodstuff for humans. This is primarily because we don't have the microbiology to digest them properly. Um, and the nutritional value is basically limited. We can't absorb very many nutrients from lichens because they have such a protective shield. However, um, interestingly, Arctic groups from Alaska to Baffin Island will wild harvest partially digested lichens from caribou stomachs to make something commonly called stomach ice cream. So they will actually find digested lichens in the wild and put a bunch of maple syrup on it, and it's a delicacy, which I find amazing. They're often baked in pits. They can be boiled to kind of cut through some of that acidity and make them more digestible. And you have to extract bitter enzymes when you eat them, usually. Scandinavians have something called bread moss, which is a flour made of ground lichens and is considered extremely nutritionally powerful. And certain masala spice blends are actually rooted in a lichenous flavor called dagapul, which is a kind of rock tripe. So if you love garam masala, the traditional form of garam masala is actually a ground lichen. And this is one of the few that you can eat without having to overly process it. And as a fun little experiment, I use the Consortium of Lichen Herbaria, which is a really neat resource online, to try to find historical examples of this garam masala lichen in Texas, and I did. And it turns out you can find it kind of everywhere. And there are only a couple of examples, as you'll see, 2 or 3 in Texas. However, if you check out iNaturalist, you'll find that they're everywhere. So you might have a spice blend growing on a tree in your backyard and not know it. And on this slide, lichenportal.org is linked. That is one of the best resources currently for lichens. There's been a huge push to digitize all of the plant herbaria in Texas and Oklahoma. And for the first time, we kind of have a historical understanding of how long certain organisms have been here. And in the case of lichens, some of these herbaria date back 100 years, 150 years. These specimens are really old. So it's pretty exciting that all this stuff is now available online. Prior to this, it, yeah, prior to this, you were essentially in the dark when it came to identification. Okay, so how do we describe them? How do we sort them? They all look really different. They're extremely biodiverse. There's got to be words and terms to figure out how to talk about them in the field. So we're going to start talking about form and function a little bit. When we see them as a whole creature, rather than as the parts that they're made of, like the fungus and the algae and the cyanobacteria and the yeast, when we see the whole thing as a charismatic creature, it's a little bit easier to identify them, interpret them. So even though one could argue that you're never going to fully know what a lichen really is unless you do genetic testing, I would argue that Charles Darwin's grandfather kind of had it right in a way. You can see the thing as its whole self. And you can see the multitudes in that whole self. But when it comes to naturalism and interpreting nature, it is extremely helpful to see it as a whole organism. So we're going to roll with that for a while. And that's how most of the textbooks and field guides are laid out. So first, I have to let it be known that many lichens look extremely similar and yet are hardly related genetically. There's also intermediary forms that are a little bit hard to categorize when they're somewhat new and young. You might not fully understand what they are, and they might actually be in the process of changing in front of your eyes. So when we're talking about growth forms, we're always talking about the vegetative body of the lichen. It's called the thallus. There are sort of things underneath it that bind it to the rock, but for the most part, we're talking about the big fluffy thing on top. And then many of them are actually defined by their fruiting body or sexual organs, like cup lichens or turban lichens. And so I have a little mock-up of the reproductive structures. There are lichens that are not always reproducing. There are some lichens that are constantly reproducing. So it can be a little bit of a toughie sometimes identifying certain ones, but for the most part, they're defined by their reproductive structure because they're reproducing all the time. So sometimes the fruiting body defines the shape and sometimes it doesn't. But most of these words to describe lichen morphology sound like bodily afflictions. I think this is one of the problems with the study of lichens, is that the words are clunky, they're strange, they sound a little bit gross. So it's kind of funny, but we have crustose, continuose, areolates, gelatinous lichens, squamulose, which are extremely beautiful, one of my favorites, and foliose and fruticose, which are the sort of more fluffy and fun looking ones that you see on trees. They're going to be larger and have a lot more flair to them. Usually, field guides are sorted by morphology and structure. So there's going to be crusts at the front and the foliose at the back, and then they'll be sorted alphabetically, unfortunately. So it's the kind of key where you have a giant book in your hand and you're standing in front of a tree and you're spending a lot of time flipping through the book, looking for something that looks kind of similar. So it's a little bit of a different identification process than you might be used to, but it's sort of a fun one too. It's easy to replicate with kids too. So the field guides that I like the best are really some of the only ones that we have. There are a couple of specific field guides to the Pacific Northwest. There's one on western and desert soil crusts, which kind of applies to Texas a little bit. But here on the I-35 corridor, there's not a lot of specific field guides for lichens. And it's surprising to me because we are actually overrepresented in a certain way. We have an extraordinary biodiversity in Texas specifically. So the fact that we don't have a field guide for lichens is of surprise to me. However, my favorite field guide right here is this textbook called Lichens of North America. It is significant. You can kill a man with this textbook. I have here highlighted all of the different lichens in this book that are in the hill country that I've seen. As you'll note, it's pretty extraordinary for being like a 700, 800 pager. That is a lot of lichens. And all of the red and orange tabs are the lichens where the photo in this field guide is a photo from my area of Texas. And as it turns out, when I ordered this book, I did not know, but when it arrived in the mail, I realized that the picture on the front is actually in the hill country of Texas, and you can see it because of the distribution. These are three that only grow on the trees outside of my house, and they're quite literally on the cover. So Texas has a lot to be proud of when it comes to lichen biodiversity, and people that study lichens are really usually focused on Texas for a lot of their studies. This book is sort of like the magnum opus of one couple. They traveled the US in a Winnebago for 10 years, the last 10 years of their lives, to make this book. So highly recommend if you're looking for like a fun Christmas gift. It's the most beautiful coffee table book I've ever had. And while it's heavy, it's extremely useful. There's also a consortium of lichen herbaria online, like lichenportal.org. It's not extensive yet, but it's a really good way to see fun stuff. This page that I have pulled up here on the PowerPoint shows that some of our favorite little historical parks have done lichen surveys. They're not super extensive, but there might be one in your area. So it's kind of fun to check it out. And I use the consortium of lichen herbaria usually when I'm not quite sure if what I'm looking at in the textbook is what I'm looking at on the tree. And so I'll try to find like a historical example that has a research grade identification. And then another really fun textbook or field guide rather is Macrolichens of the Pacific Northwest. That is a really extensive, beautiful field guide. And because it's such a foggy, humid place, they tend to have some pretty crazy abundance. And then there's also a working group on iNaturalist for lichens. It's lumped in with the bryophytes, which I don't exactly love. Lichens are not mosses, even a little bit. However, I recognize that people who might be into mosses would also be into lichens, so it's kind of the same crew. However, this working group hasn't really been super active in the last few years. I quite like it because it's a good way to kind of get a lot of research grade IDs really quickly, kind of see a diffusion across Texas, and then also it gives you an idea of key players because there really only are a few people who are studying it, and it's kind of fun on iNaturalist, my favorite form of social media, to kind of check out who is nerding out. Oh, I see in the comments it says identifying and classifying lichen reminds me of identifying galls. It's very true. Galls are extremely interesting. And also the sort of symbiotic, parasitic line that galls kind of live on is similar to lichens. It really is up to the viewer how they define them. And galls, for anyone who doesn't know, are essentially a growth on a tree that is habitated by a wasp. But to our knowledge, it doesn't seem like the wasp really harms the tree. And there are even galls in Central Texas in which there is a wasp within a gall or a wasp within a wasp within a gall. So they demonstrate something pretty similar to what lichens do in the ecosystem in terms of both mutualism and extraordinary multiplicity, let's say. So now I'm going to speak a little bit to IDing in the lab. Oh, could you go back to the citizen science that kids use in Europe that you mentioned? And is that something that we can utilize here? Absolutely. It's a 10-step process that they put together up in primarily England and Wales. That process is based on finding an area in your community and then finding a group of children who are maybe in first or second grade and doing essentially like a simple preliminary air quality lichen survey, maybe next to an area on an old stone wall, next to an area that's about to have a new road built next to it, for example. So those students would do a cursory lichen survey. They might draw some diagrams of the shape. They might measure the lichens, take as many pictures as they can, and they might even send it off to get tested genetically. And then they circle back in high school. So when they become 15, 16 years old, it's time to go back to the same exact place that they went before and to do the same survey again. And you can learn a lot about your communities in that way, but you can also learn a lot about yourself in that way. It's kind of a lovely little practice. So I'm happy to share that with the Blackland Prairie chapter at some point. I can send a little link to those materials or some delightful little YouTube videos as well. Um, so if you want to identify a lichen down to species, but you're having a hard time just looking at it, one of the best ways to do that is a chemical spot test. So like I said earlier, some lichens produce some really interesting acids and chemicals, and they have these wild kind of digestive byproducts. And the chemical spot tests are testing for those. So some of them will lack coloration in the chemical spot test. That's how you know what they are. And then some of them will change color when you treat them with certain chemicals. So that's how you understand. The difference between one lichen and one that looks very similar is usually the color change that's initiated by a chemical spot test. So they're usually using bleach. There are field-based iodine tests that are kind of fun. Um, and then potassium hydroxide, not very common, kind of hard to find. And PD microcrystals, which are kind of a funny lab invention, also pretty difficult to find. And you can also use ethanol. So this is the insanity of a chemical spot test. Some people love this kind of stuff. For me, it's a little bit, it's difficult to integrate into teaching, right? It's fun for in-class sessions, but it's a little bit hard to take out into the field because you need kind of a whole setup. But essentially, you're looking for positive reactions with color change. So there's sort of a secret code in lichen field guides and keys, which will say, yes, if it's this species of Cladonia, it would be K, P plus Y. And that plus Y means yellow, for example. So you would do a KP test, and then you would see if it turns yellow. And if it does, that's how you know what it is. So you're doing microscopic testing, but without a microscope. So an example test is kind of cool. You could test a vernia for the presence of oosnic acid. Now, oosnic acid is essentially, it's the thing that turns those lichens light green. So if you're seeing anything that's that light greenish bluish, or the usneas that hang in the Pacific Northwest down from trees that are sizable, that's going to be an indication of oosnic acid. It's a pretty strong acid, and it creates kind of bendy, like extremely strong lichens. They can be pulled on like horsehair and they don't snap. So what you would do is you would crumble up a piece of this Avernia. You would put a piece of white paper in a tiny little well or on a plate. And then you would put a drop of potassium hydroxide. So if the paper turns yellow, then you should be able to see the presence of oosnic acid. And if you drop a little bit of bleach, that yellow should intensify. So in the guide, it's going to look for the species that reads KC plus Y. Pretty straightforward. The iodine tests are the most fun in the field because they create a bright, crazy purple change almost immediately. Sometimes in order to do these tests, you actually have to use like a tiny little scalpel and scrape off tops of the thallus, like the mycelial shield, so to speak. And then you end up kind of, yeah, you dig into the lichen a little bit and you drop the chemical in there and witness the color change because you're looking for the cyanobacterial partner or the algal partner to dictate that specific test. Now, the real question is, what does a lot of this mean? Like, how do you take such an enormity of knowledge, such a crazy biodiversity, such an important part of the ecosystem, and then go into your lives and interpret it for others? The biggest thing that I tend to notice is that lichens are essentially destroyed all of the time. It's really, really common. I remember after the winter storm a couple of years ago, when we all woke up to limbs covering all of Texas, essentially, fallen limbs everywhere. It was like getting to see a portal into the overstory that we had never really seen before. And then I watched communities come together to round up all the limbs and burn them. And it was devastating. Even wood chipping them would be a little bit better. You're still doing a lot of cellular destruction, but you're at least giving the possibility of those lichens moving through the ecosystem in some way. So I would say number one thing that you can take away from this talk is that if you have a limb fall from a tree that is absolutely covered in lichens, the limb in this picture, those lichens are likely 70, 80 years old. If they're that dense, they're nearing 100 years old. They're probably almost as old as a tree if it's something like an oak tree. You should leave them on the ground. You should leave them to be essentially reabsorbed by the ecosystem, to be habitat for all kinds of microbial life, to be used by birds and nests, and then also to be slowly eaten by decomposers. Um, as well as there's some really interesting research on biological soil crusts, basically holding down the soil from being eroded away. So if you have an area that's covered in rock and limestone, let's say, and you see a lot of crusty stuff on the surface, please refrain from scraping it off or scooping it up. Those things are meant to be there for a reason. And likely they're decreasing the heat and adding habitat where it wouldn't otherwise exist. You see this a lot with power washing in the suburbs too. People just love power washing their concrete, and they don't know that their concrete is a nest of biodiversity in their area, possibly the most biodiversity in their area. So anything that has lichens growing on it, I like to leave it. I will say, I'm no fun at parties. I'm no fun at bonfires. I'll save every stick with a really nice lichen on it. But that's all right by me. They're in a beautiful stack in the woods, creating an ecosystem for birds and snakes. And I have no problem with that. So at the very least, try to relocate it before you burn it. So one of the biggest things that we could do as people who are interested in lichens, if this piques your curiosity, is we need education and outreach. We need folks who are confident interpreting the mysteries of lichen, or at least sharing the things that we don't know about them. They're completely underrepresented in textbooks and curricula, especially for school children. So that's something that could be made a dent in through Master Naturalist efforts. I will also add that when I gave this talk at the annual meeting last year, a couple of geologists approached me and they actually added lichens to the geology and soils chapter in the Master Naturalist textbook. So that was like a huge win for me. Um, and there's also no localized field guides. So if you are curious about this and you start to get passionate about it, one of the best things you could do is try to create a tiny little brochure field guide of the lichens in your area. I think making this knowledge more accessible for people and kind of compiling it, sort of giving like a heavy hitters, like the most charismatic, cool, colorful ones of the region is a really good way to get people interested in lichens who would otherwise never notice them. Or maybe they just hadn't given a name to them yet. And then there's also a general ethic or an awareness that I think you can go away with. There's this sense that fungus in general is detrimental to ecosystems. And you see this a lot, mostly in these land management contexts, right? There are actually ball moss and lichen removals that happen. There even was an AgriLife extension webpage about it for a while there, where landowners were suggested to remove these things from trees because they believed it affected the life of the oak tree. This is a misconception, and it's really best if we appreciate the biodiversity around us rather than decimating it. So if anybody around you is ever confused about what this fungus growing on a tree is, you get to joyfully tell them it's a biodiverse ancient predecessor, and it has a lot to teach them. So it's really just a mindset shift that is required. So ending this talk tonight, I'm going to leave us with some meditations and some pretty wild facts. There's a lot of little wormholes that you can go into, but I'll just leave us with a little rumination. So lichens exist on the extremist edges of our universe, quite literally. They are the only organisms to ever be sent to space because they can survive Mars-like conditions. They can survive the vacuum of space, and they can survive the UV and the radiation that the vacuum of space provides. They've survived immersion in liquid nitrogen, which is negative 195 degrees centigrade. And they're still somehow extremely slow growing. They can be extremely old. In fact, they're considered one of the only creatures on Earth next to lobsters that show no signs of cellular aging. So we've yet to see a lichen cell degenerate ever. The oldest recorded lichen is in Sweden, and it's 9,000 years old. Nobody knows when they first evolved, but we do know it's happened independently in evolution 12 times. The oldest fossils date to about 400 million years ago. That's pretty impressive to me because lichens break down in systems relatively quickly. So the fact that we have a lichen fossil is kind of a big deal. And one of the most interesting elements is that 1 in 5 species of fungi can lichenize. So there is an extraordinary diversity at the core of that. What we're also finding is that there's far less algae and far less cyanobacteria that can lichenize. So the stakes around lichens have more to do with a scarcity around algae and cyanobacteria in the ecosystem. And I just adore that they invite these flashes of insight. They're sort of a catalyst for a shift in perspective. At least they were for me. Instead of clarifying our understanding of the natural worlds, they offer a kind of mystic haze, a sort of glimpse into the wildest natures of nature, the primordial soup. They ask very incisive questions and they give many-tongued answers. What are we really made of? And what is the edge of me? Lichens are actually places where the organism unravels into an ecosystem and the ecosystem congeals somehow into an organism. Lichenologists today have no static definition of what a lichen even is whatsoever. The understanding of these organisms is in a state of eternal fluxus. We have a lot to learn from that lesson alone as naturalists and as individuals who have taken to naming things. As the amount of identifiable bacteria in lichens increases, some scientists interpret them as a microbial reservoir or a nursery for biodiversity. This is one of my favorite interpretations of all. Why did we, for hundreds of years, conceive of organisms and further ecological systems as being closed loops, as being walled off, individual or discrete? Especially when even us, our own bodies, are also nurseries for biodiversity. I would argue that Western philosophy has a lot to do with that. Perhaps the structure of our society makes it feel easier to categorize things and to name them, to define their edges or assume the edges. But lichens contain more than we will ever know. They're made of producers, grazers, and decomposers. They hold light and they hold darkness. They create and destroy. Like lichens, we too are also emergent phenomena. So I see a question in the chat. Do you think there are mucosal forms of lichenous conjunctions inside our own bodies? I think it's extraordinarily possible that our gut microbiomes house interesting symbiotic relationships. We don't really know much about it because it's such a ever-changing and liquid space. So when those things are seen, it's usually in feces and they're usually dead. That's how we know about the gut microbiome. But I would say that there's a strong possibility that there is horizontal gene transfer going on in our guts and that organisms are mutual and learning from one another in very strange ways inside of us. And the gut microbiome is still a very new field of study. It's about 25 years old, but already we've made discoveries related to human health, the brain, the heart, even our structure of hormones and feelings and everything about who we are that can be attributed to the gut microbiome and those relationalities. So it's interesting to think about the ecosystems inside of us and maybe the ecosystems on our skin. Then I see another question. If someone put some on Mars, would it continue to live and grow? So this is the experiment. Right now, it looks like they tend to go into a dormant state when they're in Mars-like conditions. But what's wild is they can stay in those conditions for over a year and return back to their ability to survive when they enter into conditions that are a little bit more favorable. So there are studies right now to essentially push the limits of what lichen is capable of to see if it can exist in Mars-like conditions. And it is kind of being uptaken by the folks who are interested in colonizing Mars as a genuine possibility for creating biodiversity there. They're even kind of trying to augment lichens a little bit and genetically modify them in certain ways to do certain jobs. So I got sent an article this week that came out from an A&M student who created a kind of concrete with cyanobacteria in it, akin to a lichen, which was able to fill the cracks in concrete by growing into them. So there's a lot of really interesting science on the horizon for this, and especially knowing that they are probably the most resilient creature on Earth. There is a lot to learn from them. And yes, Janice, maybe they already are on Mars. Maybe the origins of life are lichens, and we would just never know. Does anybody else have any questions?
Zane, I have to say, I am as blown away this time as I was the first time I heard this presentation. It was
Thank you.
amazing. Thank you so much for sharing your passion and your knowledge with us. We really appreciate it.
Oh, I'm just so grateful. I really am. It's such a pleasure. And I'm just hoping if any of y'all are interested that you bug me about it because it really is my favorite thing. And, share that curiosity with as many people as you can.
I plan to do exactly that on both counts, bugging you and sharing.
Perfect.
All right. Well, I think that's it then. We thank you very much. And that concludes our presentation.
Awesome. Thanks so much, guys. Have a good night.
Thanks, Zane.
Mhm.
Awesome, Zane. Nice job.
Thanks, Eric.
Loved listening and loved how philosophical it was.
I can't help it. I'm a philosopher.
It was perfect.
Thank you.
All right, catch you later.
Bye, guys.